Hello class. Again, I decided to break up the presentation and we'll do a separate presentation for smooth muscle. So we want to compare and contrast smooth muscle and skeletal muscle. Talk about how the process of cross-bridge formation is really different in smooth muscle. And quite frankly, there really isn't a length-tension relationship. Or we could say the length-tension relationship is always changing in smooth muscle. Smooth muscle has some interesting electrical properties. There are different ways to stimulate smooth muscle. We're going to talk about something called the basal electrical rhythm and talk about how some hormones and neurotransmitters and various things can affect smooth muscle. So here's a picture of smooth muscle from your textbook, and smooth muscle is very different from skeletal muscle in that it can contract in all different directions, which makes it very good for squeezing in the walls of organs. And so what we're going to find is that there's actin crisscrossing all over the place, and then in between are the thick filaments allowing cross bridges. Smooth muscle cells are much smaller, 25 microns in diameter, maybe 20 to 50 microns in length, so they're much smaller than skeletal muscle cells. All right, what did we learn about skeletal muscle? You release calcium from the SR, it binds to troponin, you move tropomyosin out of the way, myosin can bind to actin, you get cross bridges that are powered by ATP, and then you get this cycling where the phosphate is released, the myosin ratchets, a new actin comes along, it lets go, it hydrolyzes ATP, it resets, it binds actin again, this cycling kind of thing. Smooth muscle is completely different. Calcium comes in and doesn't bind to troponin, it binds to calmodulin. Now, calmodulin is kind of like a cousin of troponin. They're similar proteins, but a different protein. And calmodulin is in the cytosol. Calmodulin and calcium form a complex that bind to myosin light chain kinase. Myosin light chain kinase then uses ATP to phosphorylate myosin, and then that promotes cross-bridge formation. So phosphorylated cross-bridges form tension. And then we have another enzyme called a myosin light chain phosphatase that removes the phosphate so that the myosin can let go and can relax. And so you have this back and forth between two enzymes that regulate the myosin interaction. So here we go. Calcium binds to calmodulin. Calmodulin is activated in the complex, binds myosin light chain kinase and activates it. The activated complex then takes ATP to phosphorylate myosin, and then we get a cross bridge. And we get cycling of cross bridges. Now, myosin light chain phosphatase is activated with low calcium. That removes the phosphate, and then we get more of a relaxation. It's important to realize that these enzymes are working all the time, and if you have more kinase, you're going to have more contraction. If you have more phosphatase, you're going to have more relaxation. So we have two kinds of muscle. We have something called multi-unit or innervated. So your piloerector muscles that give you goosebumps. The muscles regulating the lens of the eye. The muscles regulating the iris. These guys are innervated. They need a neuron to tell them what to do. So they're not spontaneously contracting. We have a single unit or visceral. Many fibers connected by gap junctions so that one contracts, a whole bunch of them contract. And there are all kinds of things that could make these guys contract. You could have a hormone that binds to them and makes them contract. You could have a pacemaker. You could have a neurotransmitter that's released from these structures called varicosities. Some stretch can activate them. So they can be activated by a number of things. And so the gut is single unit muscle. And the pregnant uterus, so it's interesting, the non-pregnant uterus is more multi-unit, the pregnant uterus is single unit. So vascular. So after this next exam, we're going to go on to the cardiovascular system. What kind of muscle is vascular muscle? It's kind of in between. It's not so much connected by gap junctions. There are varicosities that activate it, but vascular muscle isn't as spontaneous as some other types of muscle. So vascular is in a category by itself. So it's important to realize that smooth muscle, every type of smooth muscle in every type of organ is so unique. The muscle in your small arteries is different than the muscle in your veins, which is different than the precapillary sphincters, which is different than the arterioles, which is different than the ureter, which is different than the urethra, which is different than the uterus, which is different than the gut, which is different than the duodenum, which is different than the ileum, which is different than the colon. I mean, they all have their own uniqueness. So we can say some general principles about them, but there's a lot of variability in the various types of muscle. So one way to activate muscle is through pacemaker type activity. So here you see sort of a spontaneous kind of thing. It reaches a pacemaker potential and then it has a action potential. These action potentials are calcium-based action potentials, not sodium-based. Here we see a situation where we have a slow wave. That's your basal electrical rhythm. It's below threshold. Then you apply an excitatory stimulus, you get above threshold, and now we have these calcium action potentials, and then the muscle contracts. This is very much how it is in the gut. Here shows the varicosities. So we don't have discrete synapses, but we have little bulbs that have neurotransmitter. They release the neurotransmitter, and wherever receptors are to pick up the neurotransmitter will then respond and contract. There are all sorts of chemical influences that influence smooth muscle. So lots of vascular smooth muscle is influenced by local factors. Builds up of CO2, using up oxygen, nitric oxide, buildup of potassium. Adenosine is a very important one. So all kinds of local factors we associate with increased metabolism. Epinephrine and norepinephrine. Now, epinephrine at a beta receptor causes relaxation. Norepinephrine at an alpha receptor causes contraction and vasoconstriction. Acetylcholine sometimes can stimulate smooth muscle, other times can relax it. Angiotensin II can stimulate smooth muscle. Vasopressin, also called ADH, can contract vascular smooth muscle. Oxytocin can cause the uterine muscle to contract. Serotonin is a vasoconstrictor. Histamine can relax. So how is this happening? Factors that tend to increase cyclic AMP or cyclic GMP tend to promote relaxation of smooth muscle. Things that increase calcium promote contraction of smooth muscle or vasoconstriction. Things that lower calcium promote relaxation. There are some second messengers in calcium. So calcium ion, calcium binding to calmodulin. Adenylate and guanylate cyclase tend to cause relaxation of smooth muscle. Inositol trisphosphate tends to cause release of calcium and contraction. Nitric oxide tends to activate guanylate cyclase and cause relaxation. So smooth muscle is very, very interesting in its properties. Smooth muscle can do something called a latching, where it just can maintain a contraction for hours with very little use of ATP. And supposedly what's happening is we got a kinase, we have a cross bridge, it's staying there, and we don't have the actual cycling. So I've talked as if most smooth muscle is in vasculature, and that's not really true. It's just that that's the next section that we're coming up to. In the spring course, we'll talk a lot about gut smooth muscle and airway smooth muscle and all of those kinds of things. But we have vascular coming up, and so that kind of shades the way I'm talking about things. All right, so this is our third presentation for muscle. Thanks for listening, and I'll see you in class.